Hi everyone. So today we'll be talking a bit about food microbiology and also looking at some foodborne diseases and how we contract them. So starting us off. So in food microbiology, we are concerned with all the ways that microbes can degrade food, which will lead to spoilage, and then also the ways that microbes can be used to produce food. So, examples like cheese and yogurt. And then, of course, we're also going to be talking a bit more about, as I said before, about foodborne diseases. So food microbiology, most of you will be aware that there are specific guidelines that need to be measured or met by people who produce food, the industries that produce food. And there are agencies that inspect food production or like the processing facilities where this food will be made. This will be the FDA, which is responsible for research, and it sets the standards and requirements, and it also conducts some testing. I apologize for the noise. My neighbor is mowing his lawn. And then also the USDA, which is responsible for food and safety inspection services and recalls. So moving on from that, you will note that local agencies, at local agencies, health departments typically do the inspections, and the FDA will set the guidelines. However, the FDA serves as a scientific and technical consultant to both state and local regulatory agencies by publishing what is known as the FDA Code. This sets forth model provisions for keeping food safe in restaurant, cafeteria, and institutional food operations. So if you think of something like schools, most states adopt these model provisions as legal requirements that should be applied by restaurants and food service establishment within their jurisdictions. And this brings us to the Food Safety Inspection Service, the FSIS, which is an agency of the United States Department of Agriculture, so the USDA, and is the public health regulatory agency responsible for ensuring that the United States commercial supply of meat, poultry, and egg products are safe, wholesome, and correctly labeled and packaged. So following on from there, we're going to speak now a bit for the next couple of slides. We're going to talk a bit about the hazard analysis and critical control point. So this is a program that is run via collaboration between the FDA and the USDA. And this program strives to identify points at which food may become contaminated. So something that is very important that I want you guys to note here is that this is aimed at prevention. So it's a preventative system. As the old adage goes, that prevention is better than cure. It's much better to keep your population healthy than to try and treat them once they are sick. So the basics of this program were developed by the Pillsbury Company with participation from NASA, Natric Labs, which was a part of the U.S. Army, and the U.S. Air Force for the space program in 1959. So this involves hazard analysis, which is essentially the systematic study of the ingredients, the food product, the condition of the processing, the handling, storage, packaging, distribution, and consumer use. So it's a very elaborate investigation of where potential contamination can take place. As the name suggests, you are looking at critical control points, which is any point in the chain of food production from raw materials to the finished product where a loss of control could result in unacceptable safety risks. So areas of concern can then range, obviously, from the presence of pathogens, heavy metals, toxins, physical hazards, and chemicals. And it can also include the type of treatments that the products might have received. So, if you look at something in agriculture where pesticides are applied, you don't want those pesticides necessarily to be consumed. Or, a pasteurization step, which is of more relevance to us from a microbiology side. We'll look a bit more at that later. So just to summarize, this hazard analysis and critical control points, or HACCP, is a systematic preventative approach to food and safety from biological, chemical, and physical hazards, and more recently also radiological hazards in production processes that can cause the finished product to be unsafe, and designs measures to reduce the risks to a safe level. So in this manner, this program attempts to avoid hazards rather than attempting to inspect finished products for the effects of those hazards. So you're inspecting every portion of I mean, in this case, we're looking at a seafood production where you're looking at the fishing, but the raw material, the processing, the boxing, the shipment, which will then hopefully lead to safe food if all of these conditions that the food are stored or used in are safe. So you should then note that the system can be used at all stages of a food chain, from the production and the preparation processes. So that would include packaging, distribution, and so on, and storage. Moving on, what is food spoilage? So essentially, food spoilage is just the degradation of food meant for human or animal consumption by microbes. So the source of microbes can either be natural, so this can be normal microbiota, or there can be contamination from outside sources. So if you look at animal wastes, there's a nice example we'll be looking at later on, where animal waste and E. coli is a specific problem. And then also fertilizer can of course also be a problem. Again, as I said before, fertilizers, pesticides. I do want you guys to be aware of the types of spoilage, how proteins spoil, how carbohydrates spoil, and how fats and fatty acids spoil, and what it is that the process entails. So please be sure to study this for our upcoming final exam. So how can we avoid spoilage and how can we essentially hamper the microbes? And that's all what food preservation essentially is. It's just we're making the life hard for the microbes so that we can either kill or sterilize them, or prevent or slow their growth at the very least. So again, of course, this can be done by low temperature. As you guys are aware at this point, if you decrease temperature, the rate at which chemical reactions take place will slow down, and therefore microbial metabolism will slow down. So if you refrigerate or freeze food, you will in fact be preserving them. You can also use high temperature. Pasteurization is a very good example of this. And then there's also in canning, there's a temperature process which results in 12D reduction. So essentially, this is a process where we're specifically trying to prevent Clostridium botulinum and reduce their prevalence in food sources. So it's essentially a heat treatment that will result in a 12 log reduction of this microbe and its spores. So what does that mean? Essentially, using this process, the 12D process, essentially, if you, for example, have 10 to the 8th spores in one can, if you then apply this 12D process to the cans, this reduction, this log reduction to the cans by it's a specific heat treatment where you're heating the substance to 121 degrees Celsius. So then you will reduce the number of spores to 10 to the -4 per can, because again, it's a 12 log reduction. So thus you will only have one spore per 10,000 cans. So that's a great improvement from having 10 to the 8th spores per can. You can also provide preservation by drying food sources. If you look at something like raisins, dates, prunes, or dehydrated milk, again, if you take water out of a substance, it's much harder for microbes to break it down. You should be aware of this based on the metabolism stuff we've done before. You could also smoke the food. This is something more prevalent to you guys. It's very different from where I'm from. We don't really smoke things. Anyway, so yeah, you can do that with meat or fish to then also suppress microbes and suppress microbial growth and make the food safe, preserve it. You can also use osmotic pressure. So that's something where you look at jellies, jams, and condensed milks. You can use chemicals. This can be organic acids, which are commonly used in bread or carbonated beverages, salad dressings, margarine, or sauerkraut and pickles. Remember, if you decrease the pH in these sauerkraut and pickles, you will render most microbes unable to grow and use the food. You could also potentially use antibiotics, but there are some concerns, hence all the question marks. because this could lead to antibiotic resistance, and it's just that, there's also a lot of concern on the market from consumers who don't want to eat, meat products, for example, where the animals were reared using antibiotics. So that is a bit of a concern. Sorry, I saw I missed something in the chemicals where nitrates and nitrates. So nitrite is consumed in the diet through vegetables and drinking water, but it can also be added to meat as a preservative. So the balance of using this nitrate are balanced against its unique protective effect against the toxin-forming bacteria such as Clostridium botulinum. So typically, it's thought that ingesting some nitrite is much safer than potentially allowing for contamination by Clostridium botulinum. And then you can also preserve food by radiation. Again, if you use specific radiation, you will destroy the DNA or the RNA of the microbes, and they will not be able to contaminate the food anymore. So here's an example of what the canning process looks like. So essentially, you have blanching in hot water or steam, which softens the product so that it can easily fill the cans. You're essentially turning it into a semi-liquid state, which will then, following that, you will then fill the can to capacity, leaving minimal dead space. You don't want space because that can, space is also a problem for microbes where if there's no space, they can't, grow there. Steam is then used to exhaust or drive out the dissolved air. The cans are then sealed. The cans are sterilized by pressurized steam in a retort similar to an autoclave. Remember autoclaving, what that process is, and that's how we sterilize our lab equipment. The cans are then cooled by submersion in a water bath or by spraying them with water, and then they're labeled, sorted, and delivered for sale. So yeah, that's essentially just how the canning process works. And but again, all of these processes are not really 100%. Sometimes there is still contamination that occurs if there is a weak link in your process somewhere, and it's not optimal. You still can have contamination occurring. So, for example, you can have thermophilic anaerobic spoilage, which is spoilage from thermophilic bacteria that survive in low acid canned foods. And this is often, you can see this when the can swells up because of the gas the microbe is producing. And then food will then have a lowered pH and a sour odor. You can also have mesophilic bacteria that can spoil food, especially this is prevalent in leaking cans. So this is when external bacteria are introduced during the sealing process, and this will cause putrefaction. And then most acidic foods can be preserved by processing at less than 100 degrees Celsius, but it is important to know that there are also heat-resistant microbes you should be aware of. Here are two examples of heat-resistant fungi that you then must be aware of, and you should be sure that you don't have them contaminating your foods if you don't use very high temperatures. So moving on to the microbiology of milk, this is just to give you a breakdown of what the composition of milk actually is. And then more importantly, we're going to be looking at possible sources of microbes in milk. You will remember our friend Louis Pasteur and how his pasteurization led to the revolutionary improvement in how milk and how safe milk is for use. So possible sources of microbes that we would want to then eliminate during pasteurization would be the udder and flanks of the cows, dust from the air, unclean utensils and equipment. It can also just come from humans, so workers handling the milking operation. I don't know, how many of you have been on farms or have worked from, perhaps owned farms, grew up on farms, but you know that farms are not at all what I would describe as a sterile working environment. So it is very possible for the workers to also contaminate the milk when they are actually just going about their normal jobs. There can of course also be other various factors, but these are the main ones that I want you to be aware of. So now we're going to look at the types of microbes that are found in milk. So first you have lactic acid producers. Again, I would like to note that these are non-pathogens, but these are non-pathogens. We'll be focusing on pathogens on the next slide. So this can include streptococci, lactobacilli, micrococcus, and then coliforms. Note that E. coli is here because not all E. coli are harmful. And then you can have some other objectionable non-pathogens. So what I mean by this is these aren't really organisms that will necessarily cause morbidity and mortality in humans, but they're not exactly what I would call pleasant when they are in the microbes. So for example, you have ropiness that can be caused by E. aerugines. So ropiness in milk is essentially due to the psychotrophic bacteria that is named Alkegenes viscolactis. So that's this Alkegenes species here. The psychotroph grows in milk when stored at low temperature for a long period of time. Again, remember, it's a psychotroph. And then ropiness is mainly observed when the milk is then converted into curd or yogurt. So, I mean, it does decrease the usability of the product without really making anyone sick at that point. So it's not great. Please have a look at all these other ones. I'm not going to go through them because we are a bit pushed for time. And then types of microbes found in raw milk. So now we're going to be looking at specifically the pathogens. So first is bovine mastitis, which is caused by Staphylococcus aureus and Streptococcus algalactiae. So essentially, mastitis in dairy cows is the persistent inflammatory reaction of the udder tissue. It's a potentially fatal mammary gland infection and is the most common disease in dairy cattle in the US. And it is also very, very costly to the dairy industry. In fact, the most costly. So during this, in mastitis, pathogens can be in the milk and infect other cows when the same milking machine is used. So it can then spread from cow to cow and then essentially decrease your entire farm's productivity and lead to a unsafe milk. And then we're going to obviously talk about tuberculosis, which is caused by Mycobacterium tuberculosis. So again, I want you guys to just remind you here, pasteurization was invented during a time when millions of people became sick and died of tuberculosis, scarlet fever, typhoid fever, and other diseases that were transmitted through raw milk. So pasteurization, essentially reduces this and eliminates that risk almost entirely. Now we're going to be looking at tuberculosis, which is undulant fever. So it's a highly contagious zoonosis, and you guys know zoonosis is a disease that is of animal origin that then can jump over to humans, and is caused by the ingestion of unpasteurized milk and undercooked meat from infected animals or close contact with their secretions. Then there's also Q fever, and this is spread to humans by animals, most commonly sheep, goats, and cattle. And it's caused by actually inhaling dust particles contaminated with the infected animals. So high risk occupations here include farming, veterinary medicine, and animal research. And some symptoms typically include a fever, a cough, nausea, and fatigue. And then there are, of course, also enteric pathogens, which are pretty simple, so I'm not going to break them down right here. And then, so how do we test bacteriological quality of milk? Well, there are a few options here, to verify that the milk is safe. We can look at coliform, we can look at total bacteria, so it's essentially a heterotrophic plate count. We can do a direct microscopic count if you have the time and the labor available to do this. You can also use reductase tests, which are pretty popular. This will include methylene blue and resostrin. So essentially, the principle of methylene blue reduction test depends on the fact that the color impairment to the milk by adding a dye such as methylene blue will disappear more or less quickly, depending on the quality of the milk sample that is examined. So essentially, methylene blue is a redox indicator that loses its color under the absence of oxygen and is thought to be reduced. The depletion of oxygen in milk is due to the production of reducing substances in the milk due to enhanced rate of metabolic bacterial metabolism. So essentially, the dye reduction time refers to the microbial load in the milk and the total metabolic reactions of the microorganism. And then, of course, you could also use antibiotics if you wanted to. So just to give you guys a bit more of a breakdown of pasteurization. So again, historically, Mycobacterium was what was known as the old basis. So this was the old threat that we had to make sure we were aware and suppressing in the milk. Now, in modern times, Coxiella burnetii is the new basis. So we have to make sure that the times and temperature are now sufficient to kill this Coxiella. And this is the most heat-resistant organism of public health concern. So that's why we had to update the process a little with higher temperatures so that even temperature-resistant microbes such as this can be eliminated. So first, the pasteurization process, you will have a low temperature holding area, LTH, which would be 62.8 degrees Celsius for 30 minutes. And then the milk will be moved to a high temperature shorter time, which will 71.7 degrees for 15 seconds. And then you'll have an ultra high temperature at 141 degrees Celsius for 1 or 2 seconds. And this ultra high temperature now is high enough to actually suppress well, the times and temperatures, you should be aware of both, are now sufficient to actually suppress this Coxiella burnetii too, and make sure that the pasteurized milk that comes out will be free of this microbe and it will be safe to drink. So now we'll be looking at some cheese. Cheese production, obviously also something that involves microbes. So how is that done? So it's essentially where you use milk and bacteria. So I note here that, the right ones, you can't just have any bacteria in there. So some differences here, you can use lactic acid bacteria. You can use molds, which gives blue cheese its blueness. And you can have propionic acid bacteria, which will give Swiss cheese its holes. So the bacteria that you add to the milk will alter the type of cheese that is produced. And obviously, if you use completely the wrong microbe, it will be unsafe. So don't do that. So cheese, you can also break it down further where curd, which is made of protein and casein, is formed by the action of an enzyme rennin and lactic acid bacteria. And whey is essentially just the liquid that is separated from curd. So if you stop here at curds and whey, you have cottage cheese. You can see an example there, that white cheese. I know a lot of people like cottage cheese. I'm not a fan myself. So then if you separate the two and you ripen the whey, that's how hard cheeses are developed actually. So moving on to hard cheeses, again, this is cheeses that are ripened by lactic acid bacteria. Here's the example of a bacteria specifically used for Swiss cheese. And soft cheeses are ripened by penicillium on the surface. So this can be brie, for example. Moving on to beer and wine. This is something that is usually of particular interest to students. So this is just the basic steps in the making of a red wine. So first the grapes are tested and picked, and then the grapes are crushed and destemmed. Sulfite is added to kill undesirable yeast and bacteria, and a yeast inoculum is added. Fermentation will then occur in a vat, and then the result is pressed to separate solids from the wine, and then the wine is clarified in settling vats. Here you see the wine is filtered and then aged and then bottled and sold. So here you can see the yeasts that are used during fermentation. And I want to draw your attention specifically to this area, which should look very familiar. I think back to our lectures on metabolism. And here you can actually see where how the breakdown is done and how the sugars are essentially then converted at the end of it to alcohols, which is what we are interested in during this process, of course. So here you can see beer and ale are fermented grain starches, where malting is used to convert starch-containing grains into carbohydrates that can be fermented by the yeasts. Sake, which is of Japanese origin, is made by converting rice starch to sugar by aspergillus. This is also known as rice wine. Wine is made from fermented plant sugars. The bacteria conduct malolactic fermentation to make wine less acidic. And then Acetobacter and Gluconobacter aerobically convert ethanol to acetic acid to make vinegar. So now we're going to be talking about food, some foodborne diseases. I would just like to note at this point, please also be sure to look at these chapters in your textbooks, because I had to mesh them together because we lost some teaching days because of rest days and whatever else. So I am covering this in very broad strokes. So just be sure to also look at your textbooks and make sure to study from your textbooks, prepare from your textbooks. Okay, so foodborne diseases can essentially, or food poisoning can be from two things. It can either be from infection or intoxication. So infection happens when the microbes grow and reproduce inside of your body. Intoxications is essentially when the microbes grow on your food and then you eat it. So there's the difference. So first we'll be talking about an example that is close to home for me, because there was a massive outbreak of listeriosis just after I left South Africa, the biggest one in history actually. So this is a foodborne illness that is caused by Listeria monocytogenes, which is a gram-positive rod bacteria. And this organism can cause bacterial meningitis, which is inflammation of the brain, spinal cord, and membranes, and typically follows infection. These microbes are generally, when they're phagocytized, the bacteria escapes from the phagosome and multiply within the host's white blood cells. They are typically excreted in animal feces and are readily then found in soil and water. They can grow at low temperatures. So here you can, again, they're, as I said before, they are ingested by phagocytes, but they're difficult to destroy. And they can also secrete spore-forming toxin called listeriolysin, which allows the bacterium to escape from the phagosome. And it will then grow in the central nervous system, where it will cause meningitis, or in the placenta, which can result in stillbirths. Again, here's just the meningitis being the inflammation of the brain and spinal cord that is typically caused by the infection. So this microbe typically lives as an intracellular pathogen inside of host cells, using the cell's own machinery to survive. We're very familiar with this at this point. But pathogenic strains of this organism go through an intracellular life cycle involving early stages or early escape from the phagocytic vacuole, rapid intracytoplasmic multiplication, bacterially induced actin-based motility, and direct spread to neighboring cells, in which they reinitiate the cycle. So you can just think of them as popping out from one cell, infecting another one, popping out, infecting another, popping out, infecting another. The bacterium is first phagocytized by these cells and then secretes a spore-forming toxin called listeriolysin, which allows the bacterium to escape the phagosome. And here you can see an example of that process actually happening. So as I mentioned before, there was a massive outbreak of listeriosis in South Africa during 2017, 2018. This was essentially by Listeria monocytogenes, which was in food, leading to food poisoning, and it specifically involved contaminated processed meats, which were produced by Enterprise Food, which is a subsidiary of Tiger Brands. This resulted in 1,060 confirmed cases and 216 deaths. Just to give you some follow-up to this story. So following the announcement, Tiger Brand's stock price on the Johannesburg Stock Exchange dropped by 7%. And this resulted in a $438 million reduction in total value, almost 439. But at a press conference the next day, the CEO of Tiger Brands, Lawrence McDougall, denied responsibility, saying that there is no link between any of the deaths and their products. And then when pressed by the journalist, he just wouldn't apologize. The South African government then issued a recall notice of all products on March the 4th, and Tiger Brands reportedly admitted to knowing about the presence of Listeria in some of its products 18 days before the government recall. It shows you the power of greed, right? And then on March 5th, Botswana, Namibia, Mauritius, Mozambique, Malawi, and Zambia, other countries obviously suspended all imports of processed meat from South Africa, and then Kenya followed suit on the following day. And then the latest has been that during December of 2018, the South Gauteng High Court, so that's one of the provinces in South Africa, granted a certification order opening up the process for a class action lawsuit against Tiger Brands for around 1,000 claimants. So yeah, they're going to get sued, and deservedly so. You can't, knowingly endanger the public like this. It's insane. Anyway, so tangent aside, moving on to botulism. So this is caused by Clostridium botulinum, which is an anaerobic spore-forming gram-positive rod bacteria. It's readily found in soil and water sediments. Ingesting spores does not cause the illness. However, in an anaerobic environment like sealed cans, the organism can produce an exotoxin. And this is an incredibly potent toxin. So the LD50, so the lethal dose that would kill half of an infected population, is 1 nanogram per kilogram for botulinum toxin. This is an insanely small amount, and it's typically the smallest amount found within toxicity tables. And the problem also is because this thing is just so toxic, the toxin is not present in large amounts, in amounts large enough to be effectively immunogenic, so there is no antibody production. This toxin is, however, destroyed when you cook the food thoroughly, but it can also lead to diseases such as infant botulism, where the bacteria actually colonizes the intestines, which is just severely unoptimal to say the least. So the exotoxin is specific to the synaptic end of the nerve, which blocks acetylcholine release, which is responsible for neural impulses across the synapse. Notably, the toxin is not produced in acidic food, and again, cooking or boiling it will kill the endospores, so it's not that resistant to heat, luckily. It's very common in infants, as I said before, where it can colonize the intestines, and the symptoms will include progressive paralysis. So then we can also look at staphylococcal intoxication, which is most commonly caused by Staphylococcus aureus. So Staphylococcus can cause food poisoning when a food handler contaminates food and when the food is not properly refrigerated. Other sources of contamination will include the equipment and surfaces on which food is prepared. These bacteria can typically multiply quickly at room temperature to produce a toxin that causes illness. Staphylococcus is killed again by cooking or pasteurization. And then about a quarter of people are actually carriers of Staphylococcus, so that's a big portion of the population, of course. It produces an enterotoxin, and this enterotoxin is a protein exotoxin released by a microorganism that targets the intestines, and it coagulates. It also uses coagulase, which is a protein enzyme produced by several microorganisms that enables the conversion of fibrinogen to fibrin. Like I said, this enterotoxin is very heat stable, but it's also naturally, this microbe is also naturally found in the throat, nasal discharges, and boils on the skin. Again, like I said, about 25% of the population, 25% of the population are carriers. 1 million bacteria per gram of food can actually cause illness, which sounds like a lot, but it really isn't that much. And the incubation period is actually rather short, just being between 1 to 6 hours. Very prevalent in cream-filled foods, meats, and salads. So most strains are coagulase positive, and that's how we use to ID them. Again, this is the protein enzyme that microorganisms use to convert fibrinogen to fibrin, and that's how we can test for presence of Staphylococcus aureus. So here you can just see a very rudimentary breakdown of staphylococcal food intoxication. So food containing protein is cooked, where the bacteria is usually killed. The food is contaminated by a worker with staphylococci on hand, competing with the bacteria that have been eliminated. And then you will have a room temperature holding area where this microbe will then, of course, rapidly multiply. And they will also be in the food long enough to form and release toxins. Reheating the food from this point on, so if it's sat out for a while, will kill the microbe, but not the toxin. Remember that we said the toxin is very heat tolerant. So when the food containing the toxin is eaten, and then 1 to 6 hours later, intoxication occurs and the people become sick. That's just a breakdown of how this microbe could potentially enter humans and how contamination occurs. It's important to note here that initially, when the food was cooked properly, the bacteria was actually killed, but then the contamination actually occurred by the worker who handled, or the caterer, for example, who handled the food at the event, hypothetical event, and then setting the food out at room temperature for a while made it worse, because that's when the microbe actually could multiply and get completely out of control. So now we're just going to look quickly at some salmonellosis. So essentially, this is a illness caused by salmonellas, as the name suggested, which is a facultatively anaerobic gram-negative rod. It's commonly found in the intestinal tracts of many animals, and there are more than 2,000 serotypes, 50 of which are readily isolated in the US. And there are two serotypes, 1A and 1B, with antigens 2A and 2B on the surface. So just some background here. A serotype or serovar is a distinct variation within a species of a bacteria or virus, or among immune cells of different individuals. These microorganisms, viruses, or cells are classified together based on their cell surface antigens, allowing epidemiologic classification of the organisms to the subspecies level. Just some background there. This microbe has a longer incubation time, between 12 and 36 hours. In the acute phase, you would find that about 1 billion cells per gram of feces are found, which is an insane amount. Meat products are particularly susceptible to contamination, and they can easily survive in egg albumin. Sorry. You can also, here you can see a breakdown of how the salmonella will enter the epithelials, and then will multiply once inside the cell. And then this will essentially result in diarrhea, and that's where you have this acute phase where you will have 1 billion cells per gram of feces that are present. And this diagram just gives a breakdown of the salmonella actually entering the cell and how that happens. So looking now at typhoid fever, this is caused by Salmonella typhi and is the most virulent serotype of Salmonella. Incubation for this one is quite long, it's between 2 to 3 weeks, and it is not spread by non-human animals and can only be spread by human feces. So when you think back to Typhoid Mary, we did discuss her at the start of the course. Remember that she spread the disease, she spread typhoid fever around because she worked with food and just flatly refused to wash her hands before, working with the food. So that's disgusting. So there are 21 million global cases annually, and this microbe multiplies within the phagocytic cells and disseminates into organs. So you can, especially the spleen and the liver, and when it's released into the bloodstream, and then becomes released into the bloodstream when the phagocytic cells lyse. Severe cases can involve ulcerization and the perforation of intestinal walls. And about 1 to 3% of recovered patients become chronic carriers, so they become new typhoid Marys. Hopefully now with better hygiene practices, so they're less likely to spread the disease. Again, wash your hands, people. But yeah, so once you get to have the disease, you can become a chronic carrier. So if you can, here's just a little diagram showing how we've improved healthcare, or how the US at least has improved healthcare. So salmonella and typhoid fever incidents are plotted on this graph. Here you can see the reported number of cases per 100,000 population, and typhoid fever being the black line, and salmonellosis being the pink purpley line, and then obviously time or year on our x-axis. So most developed countries saw declining rates of typhoid fever throughout the first half of the 20th century due to vaccinations and advancements of public sanitation and hygiene. You can see, again, wash your hands, and the massive decline in the transmissibility, at least. In 1908, the chlorination of public drinking water was a significant step in the US in the control of typhoid fever. And the first permanent disinfection of drinking water within the US was made in Jersey City, New Jersey, their water supply. And in 1942, doctors introduced antibiotics in clinical practice, greatly reducing mortality. And today, the incidence of typhoid fever in the developed countries are around 5 cases per million people per year. So it's really, really, really rare. Unless, of course, you Google what's wrong with you, and then you find that you, in fact, have Ebola and you have typhoid fever and everything, because, you know, Google MD, not great. Anyway, jokes aside, just a note here, you can see this almost surprising increase in salmonellosis with time, which doesn't make sense when you think about it, because, you know, as science has grown, as the medical sciences have grown, we should be more effective at suppressing microbes. But and this is a pretty strange phenomenon where we now have more instances of that. But there are a couple of factors that are that could be responsible for this, and the one at the forefront at the moment is commercial chicken production, which may be a culprit. Of course, the production of commercial chicken has just, you know, increased dramatically as the population of humans on the world, around the world, and in the US and everywhere essentially has increased so much, which just increased the amount of chicken production, which would then could then have led to just more instances of this disease being transferred to humans. Now we're going to be looking at cholera. This is caused by Vibrio cholerae, which is a curved gram-negative bacteria. Remember the Vibrio species at or the Vibrio bacteria that we discussed at the start of the semester, and how what their shape looks like. They're naturally found in the small intestine, and they produce an exotoxin, specifically the cholera toxin. And that toxin causes host cells to secrete water and electrolytes. And as much as 3 to 5 gallons of fluid can be lost in one day. Blood becomes very viscous, so it almost becomes honey-like because it just doesn't have enough water in it. So yeah, remember that the more viscous something is, the less flowable it'll be. And this will cause your organs to malfunction. You can survive in the microbe can survive in salty environments such as salt water and brackish water, and thus seafood becomes a real big problem with this. From my own personal experience, there have been quite a few cholera outbreaks, you know, back home, and I really feel bad for the people who end up succumbing to this disease, because I can think of very few ways it would be less pleasant to go, because you essentially just have uncontrolled diarrhea until you dehydrate yourself to the point of death, which, yeah, oh my God, that must just be absolute. That must be crappy, you know. So anyway, so again, the Vibrio bacteria will live on contaminated water, and then they can be transferred to various species of fish and seafood, such as prawns, oysters, and tuna. And untreated cholera can actually have a very high mortality rate, as high as 50%, but with proper care, it's less than 1%, because, I mean, if you can keep the person hydrated, that will immediately decrease their chances of death. But again, in the instances that I'm referring to, the outbreaks were typically in underprivileged communities where, you know, they didn't have access to proper health care. So, you know, and of course, with a disease that causes this level of dehydration, the longer you wait, the weaker you become. So, you know, if you think you just have a little bug and you'll be okay, because you don't have the money or the means perhaps to visit a doctor, the longer you wait, the more sick and sick you will become. And if you don't have family members or friends that can then end up helping you, you will just eventually be too weak to do anything. So yeah, really terrible. Now, moving on to E. coli. So E. coli gastroenteritis, this is caused by Escherichia coli, which is a gram-negative rod. Most E. coli, again, I want to stress this, most E. coli are harmless, but some have a mobile genetic element that can turn the E. coli pathogenic. And this can lead to things such as enterohemorrhagic E. coli, specifically the serotype O157H7. If you guys keep up with microbial contamination news, you will definitely be familiar with this serotype of E. coli, which has caused so many recent outbreaks. And then it can also have a virulence factor such as the Shiga-like toxin, which is then released when the cell lyses. Importantly here, though, cattle are not affected by the pathogen. Remember when we spoke way back when about how these diseases are not aware of what organism they're in. They can't distinguish between whether they are in a cattle or in a human. And so, they're just going about doing their thing, living, surviving, multiplying. And the sickness is kind of just a byproduct of that. It's not like the microbe has the intent of making people sick. So it's interesting here where cattle are simply not affected, but to humans this can be can cause illness. And about 2 to 3% of domestic cattle actually harbor pathogenic E. coli, but of course they're just fine. It doesn't matter to them. It has a very, very, very small infective dose, less than 100 bacteria, which this is why drinking water standards require that you have no colony forming units. You can just not have any of this bacteria in there. And then the symptoms here will also include diarrhea, inflammation of the colon, and bleeding, which you might have guessed by the enterohemorrhagic, E. coli. Hemorrhaging, of course, means to bleed. So moving on, here you can see what that O157H7 plasmid looks like. Don't worry, I don't expect you to know this. I just wanted you to show you a picture of what it looks like. Because all clinical isolates of the E. coli O157H7 contains this plasmid so plasmid 0157, and essentially the periplasmic catalase is encoded on this plasmid and may enhance the virulence of the bacteria by providing additional oxidative protection when infecting the host. E. coli O157H7 non-hemorrhagic strains are converted to hemorrhagic strains by lysogenic conversion after the bacteriophage infection to nearby hemorrhagic cells. And this can then result in destruction of the microvilli and attachment to the epithelial cells. So I don't want to brand shame here, but one of the largest cattle feedlots in the country can be found in Welton, Arizona, around 20 miles as the crow flies from the heart of leafy green production in the Gila and Dome valleys near Yuma. And there's a naturally high concentration of cattle and manure, which will then produce a fecal muddy mix in which this E. coli bacteria can absolutely thrive. And then in a dry, windy environment like Yuma, the mud can turn to dust, which can then carry the E. coli onto, products that you want to eat. So, yeah, it's a kind of a cascading circumstances that lead to increased potential for contamination. And there have been a few outbreaks at Chipotle of E. coli, which, I feel worth the risk. Their food is awesome. Anyway, so moving on to Campylobacter and gastroenteritis. So this is caused by Campylobacter jejuni, which is a spirillum. Look at the spiral shape, and it's gram-negative. This is the leading cause of foodborne illness in the US. Around 2 million cases per year is what is estimated. Nearly all retail chicken is contaminated with Campylobacter, and the infectious dose is less than 1,000 bacteria, so it's also pretty small. Symptoms include fever, cramping, and diarrhea. And one in 1,000 cases linked to Guillain-Barré syndrome, which is essentially just temporary paralysis. There's a link that you can check out. And here you can see, the Helicobacter peptic ulcer disease. You can see the formation of the ulcers. I'm not going to run through this in too much detail. We're already pushing the time here. But here again, you can see the ulcers forming and the Helicobacter present in the system. Please be sure to look at this in more detail. So if you look at Helicobacter peptic ulcer disease, this is transmitted person to person by saliva or fecal contamination. It can be treated with antibiotics and Pepto-Bismol. And this H. pylori has been linked with stomach cancer. And specifically, this H. pylori is commonly transmitted person to person by saliva, and the bacteria can also be spread by fecal contamination of food and water. In developing countries, a combination of untreated water, crowded conditions, and poor hygiene conditions contribute to higher H. pylori prevalence, and most people become infected as children, and parents and siblings seem to play a primary role in transmission. Again, in these communities, developing nations and developing communities, you'll often find that families are very large, up to like 10 kids, and they live in a very, very small enclosed space. So transmission, or the likelihood of transmission, is just so much greater because there's always close contact, and because they're impoverished. If as a parent, if your kid becomes sick, the first instinct is of course to look after them and treat them and do what you can. But that increases the proximity of these people even more, which increases the likelihood of transmission even more so, because the inverse of that is also true. When the parents become sick and there's no money, it falls to the children to look after them and to nurse them back to health. So again, you kind of have this vicious cycle of whoever gets sick will spread it to the entire family, potentially making everyone sick. So it's again also just a combination of circumstances that just lead to increased risk. I'm not going to be discussing these in any great detail. We're essentially out of time here, so just be aware that there are also protozoan diseases. There are giardiasis. There's amoebic dysentery caused by the entamoeba. Here you can see a picture of that. There are helminthic diseases, trichinosis spiralis, and tapeworm infestations. Here you can see a massive tapeworm. Oh my God! I hope that didn't come from a human. But just be sure to read up on these in your textbooks. Nothing too insane, but they are also microbial diseases that I want you guys to be aware of that can be spread by eating contaminated food. And then kind of just as a last note, we won't be having review questions today just because of how long this lecture has been. There are simply, you can think of 3 factors being responsible for contaminated food actually making people sick. And that could be temperature abuse, where the food isn't stored properly, isn't cooked properly. And there can be, or not kept frozen long enough, as my little penguin friend there is demonstrating. It can also be a personal hygiene issue. Again, think back to the typhoid people, and Typhoid Mary, how her refusal to wash her hands just, led to her spreading the disease around way more. And then, of course, also cross-contamination. Remember our example, we looked at the cooking of the food actually did kill all the microbes, but then the food was contaminated from another source, whether that be a countertop or a person actually working with the food, or a utensil that has some of the microbes on it that wasn't washed properly. There is still that likelihood that is how the microbe is transferred. So those are the 3 factors that I want you guys to be aware of. Okay, guys, that'll conclude our lecture for today, and I will see you guys in the next one. Okay, cheers.